Okay, if everyone can have a seat, we're going to get started. I'm going to move this out of the way. One side.
What'd you say?
It has feedback. When I'm done, I'm going to move it because it'll have feedback on your mics. Okay, if everyone can have a seat. We're going to get started in our afternoon session. So I have two amazing people to introduce. The first is Dr. Ed Weeber, who is the essential head of the fire initiative for the FAST fire team. So it is an amazing honor to have him here with us today. He's essentially a part of every one of the trials that we've talked about today. And the research in the mice that have been done to date and the research in the rats that are coming. So he's really an integral part of our community, as we all know, and it's wonderful to have him here. He's going to talk in the next two sessions. This one is with Agilis, which I will introduce both of them, but Ed, as you all know, is a professor, of course, and the director of the Neurobiology and Learning and Memory Laboratory at the University of South Florida. But more than that, he's a dear friend to the Angelman community, and he's a believer in our children, and that's the most important part. I'm also going to introduce Dr. Jodi Cook from Agilis, and I'm not going to call it a pharmaceutical company because they are not as well, but Agilis is a biotech company that is working with gene therapy for Angelman syndrome. And just a real brief thing about Jodi. Jodi's the chief operating officer of Agilis, and she's basically the go-to person when it comes to talking about gene therapy for our community. When Jodi and Agilis made the announcement that they were getting into Angelman syndrome, this was approximately March 2015, right? March 13th, 2015, to be exact. And the reason is because I had an alert on my email that said that something in Angelman syndrome and gene therapy happened, and it alerted me. And I found her phone number, and I called her out of the blue. I was nobody. I was a little mom with a little daughter, and she picked up the phone. I emailed her, to be honest, and she emailed me back within an hour and said, "Do you have time for a conversation?" And she talked to my husband and I for over an hour and a half about Angelman syndrome and how they have intentions to help our kids. And this is the kind of person that she is. She cares about us, she cares about our kids, and she cares about our community. So beyond what biotech can do for our kids, it's also the heart that they have that they want to help them. So with all of that said, I really am excited for Jodi to talk to you about all the progress that Agilis and the fire team have made in pushing gene therapy forward.
Thanks, Allison. Thank you very much. Okay, so am I on? Is my mic on? Okay, great. Thank you very much. I appreciate that wonderful introduction. And Allison is right. We as a company at Agilis really enjoy talking to you as families, as parents. We want to get to know you and really understand what your challenges are on a daily basis. So what I want to talk to you today about is the gene therapy program in Angelman syndrome that Agilis is working on. We've partnered with Dr. Ed Weeber at the University of South Florida and essentially brought a lot of his work into Agilis, and we're continuing to work with him to progress this. Let me tell you a little bit about Agilis for those of you that haven't met us before. We're a biotherapeutics company dedicated to advancing innovative gene therapies for rare genetic diseases of the central nervous system. So what that means is we're working in the brain, but only in rare diseases, and we're working in gene therapy. So we're trying to keep ourselves narrowly focused across different disease indications so that we can take learnings from one disease and apply it to another. The company was founded in 2013, and we're headquartered in Cambridge, Massachusetts. What I have here now is our pipeline, and what that means is these are the diseases that we have publicly announced that we're working on. Every company is always looking at multiple diseases and looking at different technologies, so we're investigating a lot of different things, but these are the diseases that we are talking about and that we are actively trying to progress. Our first disease, our lead disease, and the reason it's on the top is that it's the furthest along. And that's a disease called AADC deficiency, and I'm going to tell you a little more about that. That's in a phase 2 clinical trial that you've heard about this morning. Our second and third diseases really are for Friedreich's ataxia and for Angelman syndrome. And both of these indications are in what we call the preclinical phase, or the IND enabling. So as Scott was talking this morning about animal studies you need to do before you can get to humans, that's where we are with both of those diseases. We have another one that's more in the research phase, and this is really for cognitive disorders. We're looking at a number of disorders there, and we haven't yet picked what the lead disease is going to be. With this pipeline, you need to know that gene therapy, things you've heard about this morning, is not just a dream that's decades away. In fact, there are currently over 500 active trials in gene therapy. Now, this crosses all indications, rare and broad and cancer, but that's a big number that are going on. It's very likely that in 2017, there'll be the very first gene therapy approved for marketing in the United States. It's probably the gene therapy for the eye where they've actually cured blindness, and that's amazing. So it's a reality that can come about now. Quickly, I'm going to go back over what was already said this morning, since you all had a wonderful lunch. Let me just reiterate what we've already talked about. What Agilis works in is the AAV gene therapy, or adeno-associated virus. And this is a novel way to get a corrected piece of DNA into a patient. You start with the DNA, right? So the UBE3A is missing, or in some other diseases, there's a problem in it, it's broken, and then you get a corrected version of the DNA, and you put it in something. You put it in the vehicle, or you put it in the AAV, you put it in the virus. But inside that virus, you've already taken out all the virus issues, and you put in this new DNA. And as we all know, viruses spread very rapidly, and so that's the perfect vehicle for us to use to deliver this corrected gene therapy. We've heard a lot about safety. Well, most of the AAV viruses, all of the AAV viruses that are common, are in many of our bodies already. So they are very safe, and we took them out of nature, and that's why we're using them. The piece that we need to figure out is the delivery piece. So now we have the DNA, we have it inside the vector or inside the delivery model, and now we need to figure out how to get it to the patient. And we talked about that this morning. Do they take a pill? Is it an IV injection? Is it a direct to the brain injection? It all depends on how much you need and where you need to get it. In Angelman syndrome, the issue is in the brain, the central nervous system, which is where we work. So we need to get those cells into the brain. So as we deliver the AAV virus, it attaches itself to the cells that it's targeted to, so the neurons in the brain, and then it releases the improved or fixed copy. And as Dr. Nash was talking about this morning, we're also working on ways to, once it's there, maybe we can also make it secrete out some so it can get to the next cell and the next cell. I want to tell you a little bit about another disease that we're working on, just so you can understand that, in fact, gene therapy is a reality today. There is a disease called aromatic L-amino acid decarboxylase deficiency. I wish the person that had identified this name would have called it his own name. It would be a lot easier to call it the Highland disease. So AAD deficiency arises from a mutation in the DDC gene. In a nutshell, we can say this is the gene that makes dopamine. So you think about Parkinson's disease, old people that lose dopamine. This is children that are born with no dopamine, so they never learn to develop. Their muscles never develop. So they usually get identified when they fail to hit any milestones. Unfortunately, this is very devastating in that you really can't swallow, you can't hold your head up. So the course of their life, which is generally less than a decade, has often many hospitalizations, there's aspiration, there's suffocation. It's really a very devastating pediatric rare disease. An AAV gene therapy treatment was developed for this, and we are working on this now to take it to the next level. There have been 2 clinical studies completed, so there are 18 patients that have AADC deficiency that have received gene therapy. The first study was what's called a first-in-man study, and this is the first time, obviously, you put it in a person. So that had 8 subjects in it, and the second study was a phase 1/2. Additionally, another safety study with a little bit more efficacy. This was a single dose of gene therapy. Now, these children also have a CNS problem, so it's in the brain. The way the gene therapy was administered was bilaterally to the putamen, which is a deep brain structure. So with a neurosurgical team and a neurologist, they did mapping of the brain and then used systems that, if you've ever heard of deep brain stimulation, to target, drilled into the brain and injected the gene therapy deep into the putamen, which is really important for these children. The measurements that we took were obviously safety and tolerability of the procedure and of the gene therapy. And then we did a series of outcome measures looking at the efficacy of the treatment. So was there motor development? Was there cognitive development? One issue with this disease is there's a lot of markers in the CSF and the blood. So we can measure a lot of the neurotransmitters. So you get an indication almost immediately if the therapy actually did what you wanted it to do, even before you can measure it in the child. Also with PET imaging, you can image dopamine production in the brain. So there's a lot of measures that can be done. So that's a lot of words to say. I want to show you one of our children that we've treated. Okay, this is a 2-year-old. The top video is him before gene therapy. He is one of the youngest children that we've treated. And this was really the existence of what he could do. He could essentially lay there with a feeding tube. In the bottom panel, you can see that, I don't know if we can play it again. I don't want to mess it up. Can you play the bottom panel one more time? If not, that's fine. But in the bottom panel, they're doing a test that you do with an infant where you hold on to their wrists and you lift them and you see if they can have neck control. And most infants can have neck control. And in this case, this little boy can't. And as you can see, it's very painful. They also have severe seizure-like crises that, as many of your kids will see, these children will also do that for long, extended periods of time. So we treated him with gene therapy. 1 year later, here he is. This is at the top panel. Can you play the top panel? Let's see. Maybe I can play the top panel. I can't make it play from up here. Ah, there he is. Okay, so here he is. He's sitting up. He's moving around. I know. It gives you goosebumps, doesn't it? I mean, this is amazing. He's playing peekaboo, right? Why is he wearing a helmet? Because he's like any other toddler, full speed ahead or asleep. So they put a helmet on him just to make sure he keeps his head safe. Okay, go ahead and play the bottom panel. Now, not only that, he pulls himself up to a standing position. He's developing fine motor skills. So this is just 1 year after one treatment of gene therapy. It gives, yeah, I know. I feel the same way. It's amazing. So he has a long way to go, right? But he's out of danger. They're no longer worried he can't roll over in his crib. They're not worried that he can't eat, and so he no longer needs a feeding tube. So those really serious issues have been eliminated. Now he's in an educational system, he's in physical therapy. So we're not turning it on and all of a sudden he's at the level of a 3-year-old, but he's now able to take advantage of those services that we can provide for him. So this is really amazing work. We have in-licensed this work, and hopefully in the first quarter of next year, we're going to meet with the US FDA and with Europe and try to bring these treatments into the rest of the world. Oh, there, I did it. I made it play again. All right, next slide. So I wanted to show you that to let you know that Agilis is committed to Angelman syndrome gene therapy. We've learned a lot from this study. This is a central nervous system. This is an AAV-directed gene therapy. We have children that have had this therapy for 5 years. So it's been durable for those children for 5 years, and they continue to learn from education. So when we found Dr. Weeber at the University of South Florida and met with him and talked to him about the work that he had done in Angelman and the proof of concept animal study in the mouse, we believe that it is very likely that we can take that to the next level and get us to clinical trials with that. In additionally, we've applied to the state of Florida and have been awarded state of Florida funds, grant funds, to continue his work. So we are committed to continue that course, develop further innovative science. We have an IP position, and these are all the business issues I know that aren't really that concerning here, but we continue to work on that to make improve on what has already been done, work with Dr. Kevin Nash as well to get this gene therapy to the point that we can get it as good as possible and then move it to human trials. So for that, I'm going to let Dr. Weeber take over and tell you about the work that's gone on in his lab with us to take it to the next level. Thanks.
Yeah, it's pretty amazing stuff, isn't it? You see cartoons and you see these pictures, but when you really see what the potential is, it's a really amazing thing. I will say that I've been contacted by other companies in the past that have looked at my research, that have wanted to talk to me, partner with me. Agilis called me. I hung up as quickly as I possibly could. Somebody is calling me. And they kept up with it. They were a little bit tenacious and wanting to come down and meet with me. And it turned out that it was a good move. It's really I found them to be a different company. They're really in it to make a change. And so the partnership has been just fantastic. And Kevin Nash alongside with me, this couldn't be done without his expertise as well. So why AAV for use in Angelman syndrome? And we had thought about this a long time ago when we had done the study back in 2010, 2011. Like AADC, it's a single gene disorder. Now I know that there's larger deletions that include other genes, but you can get Angelman syndrome by a mutation of that gene, so we know it's monogenetic. It's an early onset, but it's not a brain development disorder, so it appears that all the brain structures are there, all the connectivity is there, it's just missing this gene. It's CNS specific, but it's global, which works well for AAV as well, so you need to get it everywhere. You're not just trying to target one specific area of the brain. And it's interesting because we call this a rare disorder, but in the eyes of a company like Agilis, this is coming right off of what they're saying. It's a significant patient population. So when you think about this worldwide, developing a therapeutic, it makes sense. You can treat a lot of people with this different therapeutic. And there's evidence that AAV intervention can be an effective therapy, and this was actually why they contacted me. We have done the in vivo proof of concept by looking at replacing the UBE3A gene using an AAV. So this is coming right off the paper that Jen Daly, and since Dave did it with Barbara, is Jen in the house? Is she here? There she is. So this was Jen Daly's dissertation work that she did to get her PhD. And so a lot of this was her hard work, even though I like to take credit for it. So yeah, thank you, Jen. So this portion right here, let me see if I can use this. You can see it on both sides. Perfect. So this is a cross section of a hippocampus, and you're gonna see this more and more and recognize what the hippocampus looks like. It's really a beautiful area of the brain. All of these dark areas right here, and this isn't a wild type or a non-genetically altered hippocampus. These are where all the cell bodies of the neurons kind of line up to form this structure of the hippocampus. We chose to target the hippocampus specifically because we know that there's a defect in the hippocampus in the AS mouse. We know that the hippocampus is involved in learning and memory. And we really didn't have the expertise or knowledge at that point to try to target the entire brain, but we thought we could target the hippocampus specifically and have an effect. But you can see in this wild type animal that these neurons are expressing when you stain for UBE3A, it's there. It's in all of the neurons. Right below that, this is basically a hippocampus that was taken from an Angelman syndrome mouse model, and you can see that all of that dark expression of UBE3A is missing because the maternal gene has been turned off. And this is our AAV transfected animal. So we inject this AAV, we wait 5 weeks, and then we can see that there is basically the same amount of expression in this AS mouse model as what we see in our wild type mice. So we can get expression in using this AAV. It's widespread. This was a single injection on both sides of the hippocampus. And then we look to see, what did it change? Did we recover any of those symptoms that we see in the AS mouse? I'll talk about long-term potentiation a little bit later in more detail, but this is our cellular correlate to learning and memory that we can do in an acute hippocampal slice. So we can stimulate and record and look at and measure connectivity. How well is one neuron talking to the other? And can we induce those neurons to talk even better? And in a wild type mouse, we can do this. This is in the blue up here. And when we add our AAV to express UBE3A, we recover that. Spatial memory and associative memory. Spatial memory we do with the water maze. We have a big tub of water. They have to find the platform. We recovered that as well as associative learning. So they associate a context with a shock, and I'll talk a little bit more about that in a second too. So the learning and memory, the synaptic plasticity, the hippocampal function basically came back, and the learning and memory that was associated with that function came back as well in a mouse that was an adult mouse that we injected. It had had Angelman syndrome its entire life, and we were able to recover some of those major phenotypes. That paper was great. It told us a lot about what we think about therapeutics. It tells us that we can target an adult population and recover these phenotypes. If we can do it in a mouse, then we should be able to do it in a human. But it also got Agilis interested in the research that we were doing and the collaboration that I had with Dr. Nash. So what we find is that we can get expression to be normalized. It appears that it's active. At least we are recovering the phenotype. We can recover a lot of those symptoms of the mouse. So when Agilis came to me, we said, well, how do we move this forward? How do we take this from this proof of concept into a patient population? So one thing, and we all kind of do this, if we're working in a mouse, we work with just the mouse gene. So we actually just injected the mouse UBE3A gene into the hippocampus to get these effects. So this is actually what a UBE3A protein looks like in this kind of, they call this a ribbon model of the protein. So when the protein is made, after you go from DNA to RNA to protein, it's folded in a very specific way so that it's functional. It's really kind of a beautiful protein when it's seen like this. Can anybody recognize, and I'll ask the PhDs in the group, is this rat or human? Or mouse? Yeah, nobody knows. But it did pose an interesting question, and this is another reason why it's good for scientists to partner up with somebody who's actually doing work in the human population, because we never thought about looking at this in the human gene. So we stopped everything we were doing, and we actually started looking at cloning in the human gene and putting a human gene into the AAV, which nobody's ever done before. But the question remained, are we sure that the mouse gene and the human gene are compatible? Do they have the same amount of activity? Are they functioning the same? If we put a human gene back into the mouse to evaluate it for these preclinical studies that Dr. Dindo talked about earlier, will it actually work? So they set off and they said, well, how are you going to test to see if human UBE3A is identical to mouse UBE3A? So we came up with this ubiquination activity assay. So this is a Western blot. This is a kind of a cartoon of a gel that's made out of polyacrylamide. If you put a protein in here and you give this a charge, you do electrophoresis. So this is polyacrylamide gel electrophoresis. We just call it PAGE. but it's a way to separate out proteins by their molecular weight or how big they are. And so if you were to run this UBE3A protein in this gel, you would get, you know, a big band down here where it would all accumulate. Well, what we know is that UBE3A can ubiquitinate proteins, and it actually can ubiquitinate UBE3A. So if you have one of these molecules bump into another one, you can actually get it to put a ubiquitin, if it's present in this system, on that fellow ubiquitin protein that's floating around. So what we did was we put this stuff in a test tube, and we let the proteins kind of interact with ubiquitin there, and we got different species of UBE3A. And when you run this on a blot, what you see is now these larger molecular weights will start to run a little bit different. If we do this more and more and wait over time, do this for 60 minutes, it separates out even more. If you do this for 90 minutes, you can really see where it can start getting these higher molecular weight features. So what we did was we just quantified over time, this is how much ubiquitin ligase is changing in this test tube. But does it work really with UBE3A that's being expressed in the brain? So working with Kevin Nash, we developed a UBE3A AAV with the human gene. We injected it into the mouse, and we did a couple studies. We already had the mouse gene, so we took out the protein after it had been expressed. This is actually a page that was run. This is the UBE3A, and you can see over time it's getting higher and higher in its molecular weight. This one is actually a substrate of UBE3A called S5A. But when we quantify this, what we find is that what we can pull out of the wild type, so the endogenous mouse UBE3A, or the AAV human UBE3A compared to mouse UBE3A, all look alike. So this tells us that we're using the right enzyme, and that the human enzyme is just as active as the mouse. So we should be able to put this back in the mouse and really try to nail down what the best route, the best vector, the best way that we could go forward to move this into the best preclinical trial that we can do. The other nice thing about this is, I told Agilis this is really cool. Nobody's ever seen this before. Nobody's ever done this before. Can we publish it? And they said absolutely. So we're putting this together for a publication so that the technique that Agilis has basically paid me to develop can be used for everybody to evaluate their UBE3A that they're putting in these different systems. So we did a lot of different targeting, and not to belabor the point, one of the things that we looked at was doing this a targeted injection of AAV into the lateral ventricles. So this is a cartoon of a human brain. In all of our brains, we have these kind of empty areas that are called ventricles, where cerebral spinal fluid flows. And so instead of trying to do multiple injections all in the tissue, what we can do is we can target the lateral ventricle and then let the normal flow of CSF kind of bathe the entire brain with the AAV. So we don't have to do multiple injections. We could just do this one injection. This is very simple. You're going through a very thin part of the brain to get to the ventricle. And so this is what we did. So we started to ask the question, using what we had done before with direct injection in the hippocampus with the mouse, can we see differences in lateral ventricle injection using this human gene therapy? So this is another cartoon for those of you who don't know what hidden platform water maze is, just so that when you see it again, you'll know what I'm talking about. But this is done by having a big tub of water. We put in coloring so that it's kind of milky color on there. And then we have a platform that's in a certain area that's just under the surface of the water so that the mouse cannot see where it is. And then we put visual cues around that water maze. And on the first day, the mouse just is swimming around. If it happens upon the platform, we leave it there for a little while so it can orient itself to where these spatial cues are. By day 3 doing this 4 times a day, by the 3rd day, they're getting pretty good at it. Okay, they'll swim around, they'll remember where those spatial cues are, and they can identify where the platform is. After 5 days of doing this, doesn't matter where you put them in the platform, They recognize where they are, they'll go right to the platform. Okay, so it takes a little bit of time for them to learn this, and we can measure what the latency is to find this platform. So then we do a probe test where we actually take the platform out, we let the mouse swim around, and then we say how much time did it spend crossing over where that platform used to be. Okay, so this is really the memory test. Okay, so we're training them here to make them learn, and then we're making them remember later on down the line and see and quantify how many times they go over that platform. So this is the results of that lateral ventricle injection. Latency, we've seen this before, that the Angelman mice do pretty good in the learning part, but what they don't do well in is the memory part when we do this probe test. And so when we remove the platform and we say, okay, can you find that platform now? 72 hours later, these are called heat maps where we put all the animals together. The red portion is where they spend the most amount of time, and then it goes yellow, then green, then blue. This is our lateral ventricle injection UBE3A, and you can see they spend a lot of time around where the platform used to be. These are just injected with a green fluorescent protein. The Angelman syndrome mouse don't do as well. And then this is our wild type. So the wild type and our injected mice look identical. So we could recover that spatial learning defect. The other thing we wanted to look at was specifically in the hippocampus. Now remember, we didn't inject the hippocampus, but can we have an effect in the hippocampus the same way we did if we injected directly into it by using this lateral ventricle injection? And so we look at this long-term potentiation, which again is our kind of experimental model for looking at learning and memory. And it simply measures how well two neurons can speak to each other. And indeed, this is our Angelman syndrome mouse that was injected with GFP. We give high-frequency stimulation. Those neurons, after a while, they go back down to if they've never seen that stimulation before. But our LV-injected Angelman syndrome mouse with the human UBE3A gene comes right back. So we can target the lateral ventricle. It appears that it's getting everywhere, and it's sufficient enough to overcome not only the spatial learning defect, but the synaptic plasticity defect that we see in the hippocampus. Pretty amazing stuff. So, just in summary, our ubiquination assay is telling us that the human gene is really the way to go. We're not going to move forward in preclinical studies or use the mouse gene to inject patients with Angelman syndrome. We're going to use the human gene. We know it's the right one, and we think that the lateral ventricle injection site is a really good way to get that AAV in and bathe the brain, and we know that we can recover a lot of the symptoms in this mouse model. There's still a lot of work that we still have to do to really kind of refine this. Will smaller doses work? Do we have to worry about having larger doses of AAV or larger expression of UBE3A? We're in the process of figuring that out. And we're really trying to refine what this vector looks like, how all the parts of the AAV, and look at these different types of injections that Dr. Nash talked about earlier to really get down to what is the best route, what is the best looking AAV, and what that AAV contains to get the best result possible. And so this is what we're doing with Agilis and partnering with Agilis right now. And with that, I'll hand it back over to Jodi.
Thank you very much. And this is a great partnership, and a lot of great work has happened this year. So we feel very confident that we want to move forward with AAV gene therapy with the UBE3A, the human version, obviously, and now we're doing these tweaks to get it to optimize the various components of it. When you take a drug, when you do the drug, if you think about the drug development process, when you take something into a human, you have to call a freeze. There has to be a point where you freeze everything, which means you cannot change anything. So we're working on that point to call that freeze to get that final therapeutic, and that's what we're going to take through the whole process, because you have to have one set treatment that you're going to move forward. So everyone's already talked this morning about drug development process and the different steps, so I won't go through it again. Just to say that there's a series of work, and I like to think about it, there's a series of work before the human, which is a lot about what's been going on in the Angelman community over the last decade. And then there's a series of work that will go on once we get into the human, and I think Ovid's going to be the first one to get there, hopefully by the end of this year, if not in the very beginning of next year. So as a community, it's very exciting, I would imagine, for you, because you've been in the pre-human side, and now you are moving into the human side. So it's really exciting. The things that go on the before human study side are the discovery and the basic research. There's a lot of things that have to happen and have to fail before we can get to the right answer. And that's just the way it is, right? We have a bunch of ideas, you try them out, many of them don't work, until and a lot of times you end up stumbling on what's really the best idea. So a lot of that work has happened, and what we're going to be doing right now with Dr. Weeber and Dr. Nash is the tweaking of the final design that we want to freeze. Then we want to take it into a larger and a larger animal, as Dr. Dindot said this morning. We're in the mouse, and as he said, it's kind of obvious, and you shouldn't really even need to say this, but a mouse is not a human. A little bit of injection in a mouse brain, you get the entire brain, nothing like a human brain. So we want to go up into the rat and probably up into the pig, most likely into the normal pig, and that would be mostly the safety study. Even though in our children with AADC, we don't have any safety indications with AAV, it's a different population, it's a different gene. So we want to repeat that, and we will repeat that in a larger animal just to make sure things are safe. So that all happens before you get into the human studies. Once you're in the human study phase, it's not just a matter of treating and looking for all the outcomes and finding something that looks good. You have to make a claim. You have to say, when you're looking for approval of a drug, you have to say, my therapeutic will do this. And then you have to set out and prove that. Even if you prove something else that you didn't know, you have to target what you're going to prove. So you may not get a drug approval if it gave a lot of benefit, but you didn't claim that's what you were going to test. So there's a lot of work that Ovid's been doing up until this point. You wonder why you do all these different things, because you really need to understand the mechanism of your treatment and what it is you're trying to test and what you want to prove. And as you go through the clinical trial process, you learn that more and more. In that, to get to that point, we want to understand what happens at the baseline, what happens at the natural history level. So a lot of things that are going on in the community, a lot of testing that's going to be coming up, has nothing to do with the therapeutic, but it's so when you get a therapeutic into that point of a final clinical trial, we know what happens at the baseline. So we have something to compare it against. And that is the second half of that drug development process once you're into humans. And I'm talking about in a patient-centered, what now we refer to as patient-centered drug development. Essentially, that's looking at what's going on currently and applying, we know what it is, and can your therapeutic make it better? We've had input from the community. The first thing we did when we came out into, Allison reached out to us, but we had already talked to Paula. So we want to understand the issues that are going on, particularly in rare diseases. And I think every organization, in fact, they're all here. All the organizations that are working in this area are all here meeting with the families and the patients. We can't, with a small group of people with a rare disease, it's not right to just randomly throw out a trial and see what happens, because we don't want to use patients as figuring it out. That's why everybody's here in the beginning, understanding your needs, understanding the issues, and looking at a therapeutic to address one of those certain issues. What we've started over the last year is an organization of scientists and clinicians, all the partners in industry that are interested in Angelman, and we've gotten together, and Allison mentioned this morning as well. We've now gotten together 3 times. It's called the ABOM, which really looking at Angelman biomarkers and outcome measures. And this is, we're looking at things that we can use to test in clinical trials. This organization is being run by Terry Jo Bichelle. Thank you very much. Oh, Dr. Terry Jo Bichelle, she just finished her PhD. Congratulations. And so now she has plenty of time, as she would not, I don't know, I can't see her, so I'm sure she's not there she is in the back. Now she's putting full-time effort into moving this forward and is doing a great job of getting us all together. This is something that we can do in a pre-competitive space, which essentially means all of us that are in the business side of working with Angelman can collaborate and work together to get these measures, because we all need them. Everyone needs them. No one's going to win on, ooh, I developed an outcome measure. We all want to get a therapeutic approved. So when Dr. Weeber was asked Agilis and said, hey, can we publish this data? Absolutely. And I know that everyone else in the field feels the same way. If we develop something that's based on the science or the understanding of this, then we want to move it forward and we put it out there so everybody else can use it, because we will be successful if we can collaborate in this early phase. Then as different organizations have different therapeutics, we can all compare them against the same thing and see what might work. So one company's already moving into the clinical trial phase. Others will be coming along. So the more information we can have, the better. So what is Agilis? We've been in the field now for a little over a year. We have filed for and received Orphan product designation in the US and the EU. And what that means for us, there's some financial incentives there, but there's also some regulatory guidance, which is really important for a smaller company. We've developed and tested the human gene. We've evaluated several routes of administration in the mouse model, and we've really spearheaded and got A-BOM up and running, and now we're happy to have Terry Jo take that on full time. Our next steps is we're going to investigate an optimized route of administration of gene therapy. So we've been doing it in the mouse, and that's a relatively fast way to do it. We need to move to a larger animal to get a better idea of how the therapeutic spreads. Particularly when you're going into the central nervous system, there's only so much outside material you can put in, right? You couldn't inject a bucket full. So we need to figure out where it's going to spread and how to make it spread and how to target. Then we're going to look at safety studies. And this is a requirement for registering with any regulatory body, and it's a requirement that we all believe in, that we want to give enough of the treatment to a large animal and then wait 6 months and just make sure that there are no unforeseen side effects there. The other piece that we don't talk about a lot at these meetings is the manufacturing process development. This is an area that is really critical in that we want to be sure that every time we make it, it's exactly the same. So we have to put processes and controls in place that every single dose is exactly what we believe it is. So this takes several years to develop, and we are in that process, and it's an ongoing process. Finally, our next steps is the phase 1 clinical protocol. We're not there yet, but we want to, and we have started to talk to clinicians and investigators about what should that be. We know phase 1 is always safety, but just like our kids in AADC deficiency, we want to do a series of outcome measures as well to start to understand what it is we want to study. And claim as our, what is our one claim that we say our therapeutic will do. Age comes up all the time. What's the right age for gene therapy? And what's the right age to start with the first in-human study? And that really, from a regulator's perspective, it's a risk-benefit profile. So for our children in AADC, that generally their lifespan is less than a decade. They were, the risk-benefit profile changes whether you're going to do a direct brain injection or not. One thing we've learned from that study is we've treated children from age 2 to 8. All children at all ages have received some level of benefit. The younger children have received more benefit. So it just, I would say, from a theoretical perspective, younger is better, and I think everybody would kind of agree with that. So it comes down to where do we start, how do we start, we can figure that out, but we're certainly going to be looking at in all age potential population for starting. And then obviously we're going to have the regulatory discussions. And I will kind of end here on just a couple quick questions that I know will come up. I don't know. The answer really is I don't know if your child or family member will qualify for the trial. It ultimately will depend on those inclusion and exclusion criteria. And again, I don't know when it will become available for clinical trials. And that depends on our regulatory interactions. We have to do the 6-month large animal safety study. So no matter how much we want to go faster, that is a guaranteed amount of time we have to take, in addition to the optimization until we call the freeze on the therapeutic. And so what can you do today? You will hear at the end of today from Megan, who's going to talk about the worldwide registry, which is going live today. I'm sorry if I stole your thunder. You can join that. You can participate. You can keep the records, keep good medical records of your family members. Log in, join the registry, and communicate with your advocacy group just to keep yourself informed of what's going on. So when more and more therapies and treatments come available or more sites open around the country, you're aware of it. And you can make the decision, is it good for you to join the clinical trial? If you don't join the clinical trial, if it doesn't work for you, that's fine. That doesn't mean that you're not, it doesn't mean that you're not going to have an opportunity to have the treatment if it's successful. But you can, from home, join the registry and participate. So the last thing I want to say is that we are a collaborative company. We cannot be successful without collaboration. That's what we have with Dr. Weeber and Dr. Nash in the business relationship, but more importantly with the family and the friends, the FAST organization. We can't do this without your help, and we want to do this, but we are here. We want to talk to you. Don't be afraid to come up and ask us questions. Mark Pikett is the CEO of Agilis, and he is coming in tomorrow morning. So please find us tomorrow Saturday. Find us at the ball. Come up and introduce yourself. Show us pictures of your kids. I share those pictures that I get with the rest of the extended team. So when we're working late one night and really don't feel like working anymore, we just bring up the videos and the pictures of your children. And I promise you that we are fully committed to doing whatever we can to help find a treatment for Angelman. Thank you for your attention.
What time?
You want to do, oh, we have time for a few questions.
I'm sorry. Can I just ask one question?
Sure.
You were talking about, you showed the beautiful video of the little boy who was rescued by the gene therapy,
right.
With the AADC, was that
yes,
disorder?
That's correct.
So when they're rescued by the gene therapy, and you said that they don't instantaneously become normal, so to speak, right. Are they able to learn at a faster rate than a typically developing child because they're sort of catching up?
Right, that's a good question.
Developmental age?
Yeah, that's a good question. I don't know the exact answer to that. I'm going to say not necessarily. So in these kids, it turns on the protein dopamine and some others. So in Angelman, we would say we would want to turn on the E6AP protein. The UB3A would turn, right? Mm-hmm. Yeah, so in this, no, I would say it doesn't necessarily mean they'll go faster. We're seeing a variety of experiences and progress, and then there's also a variety of educational settings as well. So it's hard to know if it's the gene therapy that's different, if it's the age of the initial treatment, or if it's all the other factors involved in it. And a lot of these children were also very sick when they got treated because they were pretty severe. So there had other complications medically that may also play a role in that. But they all have benefited from levels of therapy. They all had therapy. So, we did not do the case, there was no case where you did gene therapy and then you just watch them. But they are learning.
Thank you both very much for your presentation. It was tremendous. That example of the success with that AADC was fantastic to see. Very scientific presentation, so at the risk of sounding like a basic question, I'll still move forward. A little clarity here. So when you do an injection, are you injecting a gene that would replace the deleted or mutated UBE3A, and then it proliferates from there to create normalcy, quote unquote? Is that kind of the gist of this? Am I catching that right?
So this is something that Kevin actually didn't talk about when he was giving his presentation, but no, it's not targeting the site of where the gene was. So viruses are just good at putting in whatever DNA they have in there. We've put in the UBE3A gene, so it will insert it in a random place. But along with that gene, we have the promoter that Dr. Stiegel talked about. So we have all the mechanisms in place to turn that gene on once it gets put into the genome. Does that make sense?
Yes.
Okay. Any other questions? All right, thank you.
Thank you again.
All.
Hey, Dr. Weeber? Just a quick question for you about the FDA collaborating with any other worldwide agencies. Is there other groups or administrations that they're working with to collaborate on, a worldwide effect versus just a federal effect?
As far I know that the FDA and the EMA have had recently working groups monthly to try to collaborate, if that's what you're asking, on on the regulatory side of things.
Just on a global
and to some level, it's still independent countries have their independent regulatory authorities.
Sure.
But I do know that EMA and FDA in particular, and in particular gene therapy, have started a monthly telecon to see if they can start to kind of agree that way. I also know that some other countries, if you get US FDA, sometimes when you go to other Asian countries, they look at that as a potential positive, and so you may not have to go through a certain level. You may be able to go through different steps there, not the full-blown package. And the same with EMA and FDA. You have to do the complete filing across for those two groups, but a lot of times you can get consultation before you're totally finished with one, that you don't have to repeat things for the other.
Sure. Okay, so it is group effort globally.
Yeah, group effort globally would be great, I know. We would like that too as companies, to make one application and have it accepted, but.
Is there any scientific possibility, I guess, of like a donor program, similar to like an organ program, for getting the UBE3A from a donor and then injecting it directly human to human? Has that ever been thought about, or
it's really
kind of bypassing?
Yeah, I don't think that we need to do that.
Yeah, it's really unnecessary, because what we're doing with the virus is we're making a lot of viruses that have the identical gene in them. So we can clone the gene in. We make sure there's no mutations. We make sure that everything looks good, and then we just make a lot of those viral particles to be put in. So anybody who received AAV therapy would have all the identical gene. Okay. There's really no reason to do anything different. It's easier to grow up those genes and replicate that gene over and over again to package into the AAV.
Okay. Appreciate the time. Thank you.
Thanks.
Can I ask one question? Dr. Weeber, is there anything as parents that we could do to expedite this? Such as, you said you were getting state funding from Florida, and I happen to live in Tallahassee. Besides donating to FAST, is there anything that we could do as individuals in our state to come up with? Do I have to go to the Capitol and meet Rick Scott with my daughter and buddy up with him? Because I have a flaming hot passion of dislike for him, but I will do it. Marina and I will become his best friends, because what funding from the state of Florida would help to bring this quicker to all of us?
Well, I'll tell you, I think politically we see the same thing, but scientifically, they've actually put this really nice thing in place, and what it's supposed to do is it's supposed to increase businesses coming to Florida.
Well, that's what I wanted to appeal to him.
Yeah, well,
for.
they already have that in place, and we're actually doubling the amount of money that Agilis can bring in through those state funds. So it's great. We're getting twice the bang for their buck.
Would it help if he met? There's at least eight that I know diagnosed in Tallahassee. People from 40 to like 5 with Angelman's in our little teeny burg up there.
Well, at least the ones from 18 to 49 should contact me. So.
Okay. One of my proudest days, we were at his inaugural parade, and Marina was sitting there, and one of the aides went, "Look, handicapped photo op," and they came over and to shake hands, and Marina was drooling down, dripping from her elbow, and totally grabbed him, my hero, Rick Scott, and slimed him from ears to elbows. And then that night was violently ill, and everyone that worked for the state of Florida was like, "Yes, Marina," and waiting to see that he canceled things. But I can put all that behind me.
I hope
it would help.
Yeah.
To make some contacts.
It actually is a very, very nice program, and Jodi will probably attest to that.
Okay, well, let me know what it is. Seriously,
yeah,
I'm not.
Well, it is a corridor program to when a company comes in, if they start to put a footprint in the state of Florida, Florida will match whatever funds they're putting in, and we're utilizing it perfectly. But we're here for FAST, and FAST is also partnering with Agilis to help us do the research as well. So this is, it floats all boats. It really does. But the state is helping us out quite a bit.
Well, I'd love
and
to
so
put
is the
Angelman's
university.
on their radar.
Yeah, well,
All
you
right.
can go in and slime as many
Slime the
politicians as
hell
you
out
can.
of them.